morning. I want to thank you for having me be part of this group and talk. And also, I think the order of the presentations is really awesome because in my talk, you're going to see kind of like I'm going to dip just very briefly into a lot of other areas that previous presenters have gone in depth in. And the general thing that I wanted to try to express in this talk is not only to give you an idea of the work that we're doing in our group over at MGH, but also kind of how we got to where we are and that thought process and how I think that can be applied to other diseases and a lot of diseases that we're talking about here. So we have to start with a little bit of a history lesson, particularly with where we started with Alzheimer's disease. And as it stands right now, we think of Alzheimer's disease as, in the research, as kind of split between two different ages. It began with the discovery by Dr. Alzheimer's himself all the way back in 1906. And the pathology related to the disease was very upfront and apparent. You have amyloid plaques and neurofibrillary tangles. Obviously, it would be many decades before they would piece apart what those were made of, but they were very easy to see underneath the microscope. And even all the way back in 1906, there was an idea that this may be infection related. But around 1984, the story kind of changed. And the shift was that we not only started getting into the age of genetics, and we found the APP, the amyloid precursor protein, in the genomic sequence, but we quickly isolated the amyloid peptide tau as being part of the tangles, followed from that. So now that we had identified what these pathologies were, okay, well, the next logical step is we need to get rid of them. So that kind of began this multi-decade process of the idea that we have amyloid. Amyloid's not supposed to be there. Amyloid leads to tangles. Tangles aren't supposed to be there. Tangles lead to neuroinflammation, or amyloid leads to neuroinflammation along with tangles, and we get Alzheimer's disease. It became known as the amyloid cascade hypothesis. This is what everything has been based on. All the recent drug trials that have failed have been based on this model. We see the peptides we don't want, we get rid of said peptides, that should cure the disease. Well, it obviously hasn't. And the byproduct of that failure and that little bit of the blinders up on people focusing specifically on this amyloid cascade hypothesis has been that most of the pharmaceutical industries have ceased research into Alzheimer's disease. Within the last 5 years, basically your big three pharmaceutical companies have shut down their Alzheimer's research divisions, and I've seen an importing of people from industry back into academia as the industry's no longer interested in it because they took their shot and it didn't work, and that was billions of dollars gone. But it's not without reason why they took this path. Here you can see that from about 1980 to onward, I say within the 2000s, there was an idea of this is what the amyloid peptide does, and it's a mistake. The body creates it. It's not supposed to be there. There are other diseases out there where a mutation occurs. The mutated peptide or protein is expressed, and that causes the disease. So that was the assumption. But as time has gone on, we've seen that a lot of these predisposed ideas about amyloid actually turned out to kind of be wrong. And it took looking at other peptides to realize that, intermembrane cleavage, that's a normal thing. Peptide only generated in the AD brain, well, it's actually shared amongst all types of animals, and the A beta 42 peptide, if we look at other animals, is ridiculously highly conserved. There's a fish called the coelacanth that is kind of called the dinosaur fish because they consider it to be relatively unchanged for about 400 million years. Its sequence for A beta 42 is actually identical to ours. There's no difference between the two. And again, fibrillization, the idea of peptides sticking together. You just heard about nets. I mean, that's a little bit related in the idea that fibrillization plays an important role and can have normal activities within the body. So given that information, and as those predispositions have kind of fallen by the wayside, our group said, well, wait, if we look at antimicrobial peptides, which are peptides that are known to be expressed as part of the immune system and are part of the innate immune system, so it's a front line of defense. There's one particular one that we focused on called LL37, and we started realizing that some of the structures and interactions of the peptide, amyloid peptide, and LL37 were very common. And on top of that, we can't ignore the fact that AD has been associated with pathogens for decades. The first look into HSV1 virus in the presence of AD goes all the way back to the late 80s. So basically, right after we discovered what the amyloid was made out of, we started looking at, well, is the plaques, these amyloid plaques, made solely out of just the amyloid peptide, or are there other things in there? And there were other things in there. I mean, this is just a cross-section of a handful of different pathogens, but it's actually been connected to many more other pathogens, and they have identified them in plaques, as well as in the brain and the surrounding area of AD patients. So that led my former mentor, Robert Moore, to come up with this hypothesis that the amyloid peptide is part of the immune system. It's a natural function of the human body to produce this peptide. And the way that it would work is that the amyloid binds to glycoproteins on the surface of a pathogen. It can prevent that pathogen from interacting with the host cells or preventing the infection. And then it starts to agglutinate and entrap these pathogens into essentially what becomes a plaque. And the first target's the easy target, bacteria. We know the structure of the outer cell wall. We know what's out there. And they ran a very basic experiment where they just put synthetic amyloid peptide in with golden staph. And when golden staph gets stressed, it turns gold. Well, it did. And when they put scrambled peptide in there, it didn't turn gold. So obviously, the peptide was doing something that was stressing out the pathogen. And we took that one step further and we said, well, if it works for bacteria, it's probably going to work for viruses too. And viruses posed a more interesting relationship to Alzheimer's disease because you're more likely to see a virus running around in the brain than you are a bacterium. We know viruses can get in there. Herpes virus is one that we know goes into the brain and kind of sits around and gets into this idea of a chronic infection. Bacterium, less so, not possible. There is chronic infections that are related to bacterium and fungus. You're more likely to see a little bit of that encephalitis and a more acute response. But envelope viruses like HSV1, again, have glycoproteins on the surface. And we ran kind of the same hypothesis of binding to the outside, preventing the infection, and then sticking those viruses together. And yeah, well, we dove straight into animal models, and we looked at, you can see we have in vitro culture here with Candida, Salmonella. We have worms where we infected them and then looked at their survival. We have Drosophila, and then we also have mice. And we found that in both with bacterium infections like Salmonella, as well as HSV1 infections, those mice that expressed, or those worms that expressed, or those cells that express more amyloid survived better. So there was a protective effect. When we dove down deeper to look to see what was happening, we saw this idea of agglutination, and that within 15 minutes you can see this is the viral particle here and the outer surface of the envelope, and you can see this kind of tail that's coming off. That's actually amyloid. That's the beginning of an amyloid fibril. So it is attached to that seeding point, and then it started to string off. And then as it grows, it starts interacting with other amyloid, other agglutinated viral particles, and essentially you start kind of forming this mass. And we saw the same thing with salmonella. And when we went back, you know, we did animals, then we went super in vitro, then we kind of went back towards more complex cellular models. We used a 3D culture model with human tissue because we wanted something that was going to be a more accurate representation. And while the 3D culture system it's okay, it has neurons, it has oligodendrocytes, it has astrocytes. It doesn't have some of the heavier duty immune cells. But even without those present, we were able to see HSV1, HHV6 cause a response and cause the agglutination of viral particles with amyloid. And this video that you see here on the right, that's actually a cellular-free system. All that was put into that was amyloid and HSV1 and a matrigel matrix, a gel matrix. And the matrix provided the scaffolding, essentially, for a plaque to form and the A beta to agglutinate the viral particles. When we went back into those mice that we examined and infected and survived, we actually saw a very common morphology of the amyloid plaques, these kind of dense core plaques that you can see where there's significant amyloid on the outside, but then it changes on the interior structure. And co-localizing with those amyloid plaques was HSV1. So, you know, with this research, we kind of started answering a couple more questions and veering away from this idea that amyloid is not supposed to be there. We see this response, that amyloid responds to an infection, and that the response, even in overexpressing cells, is more aggressive in the presence of an infection. We see the microbes starting to act as a seed for amyloid plaques, and that amyloid will start to, eventually, if you leave amyloid by itself and there's a high enough concentration, it will form a plaque on its own. But at lower concentrations, microbes provided a seed for it to start forming plaques. And in the 3D culture systems, we were seeing plaques stimulated by pathogens 2 to 3 weeks before those same cellular systems would form plaques on their own. It's diverse. It's not just focused in on one virus or one fungus or one bacteria. This amyloid can interact with multiple different pathogens. So we built a case for ourselves to say that amyloid is involved in an infection etiology of AD. But that data was just kind of a little bit of a surface coverage of where amyloid and this story has taken us. Beyond being able to entrap the pathogens, we've started to see a case that the amyloid can also kill the pathogen. So once it's trapped in these nets, we start to see death of the bacterium that are in there as well. Beyond that, we have, I believe it's psoriasis, is a role where the antimicrobial peptide kind of has flipped, and it's more of a problem causer than it is a problem solver. And we don't necessarily know where that fine line is with amyloid yet. But in this experiment, we had cell cultures. We applied either synthetic amyloid or cell-derived amyloid, and we didn't really see death of the cell culture. When we add an infection like HSV1, We saw some cytotoxicity, which we expected. But then when we have both this infection and the amyloid present, the death of the cell is accelerated. And you can see that kind of in two different lights. You can see that as a problem or a solution. The problem could be that the amyloid is interacting in an infection environment and it's starting to damage cells, or we're actually targeting cells that have been infected for cell death to try to cap the infection. Basically, the agglutination process at that beginning layer failed, and we didn't prevent the infection. So now how do we treat the pathogen process? And it may involve killing the cell. But the story, as I pointed out earlier, goes beyond just amyloid. We know that there's tangles and neuroinflammation are very important roles in the etiology or in the pathology of Alzheimer's disease. Well, tangles aren't excluded from this story at all. Tangles are formed by hyperphosphorylated tau protein, and the tau peptide acts normally as a microtubule stabilizer. And when it gets phosphorylated, it dissociates from the microtubules, and then you actually start getting a breakdown of the microtubules. Well, why this might be important is because when we think about viruses in particular in HSV1, HSV1 has a really nifty way of avoiding a lot of elements of the natural immune system. And one of the ways that it does that is that it can infect a synapse and then use the microtubule network to move retrogradely to the cell body. So not having to be outside where a lot of your immune cells and your antibodies and both your adaptive and your innate immune system are freely flowing around looking for targets. It's hiding inside the cell and using that traffic system. When we take a close look at these cells, what you're seeing, one of focus is on the right here. The blue is hyperphosphorylated tau and the red is HSV1 particles. And the interesting thing, the pattern that we're starting to see is that not only do we see a grouping of hyperphosphorylated tau and HSV1 in dystrophic neurites, or kind of like these little spheres that you can see here, but we're also seeing a significant upregulation of phosphorylated tau in neurons that have viral replication centers. So all of these extremely bright red dots that you see in the soma of the cells, those are HSV viral replication centers. So not only have we gone through the infection process, but now we're to the viral replication process. And there seems to be a response based on that. And if we're breaking down the microtubule pathways that viruses can run up and down on, then we're adding a barrier towards the spread of the disease. And this plays into here with looking at how the phosphorylated tau could play as almost like a barrier action. And what you're seeing here is a microtubule system with 3 chambers, and we've seeded the 3 different chambers with 2 different types of cells. So the G10 cells are naive cells. The A4H1 are amyloid overexpressing, because we know overexpression of amyloid leads to tau pathology. And we infected the chamber on the left here, and we have microtubules that interconnect the chambers. The microtubules are just big enough for the processes to pass through, but not big enough for the cells to actually intermix. So the thought process of this experiment was we can somewhat monitor the movement of HSV1 from one population to the next. And this was a very surprising result in that not only did we see an increase in phospho-tau, but the phospho-tau increase seemed to be independent of amyloid. And on top of that, the strongest phospho-tau expression was in the microfluidic channels where there was that initial connection between the infection, the infected cells, and the uninfected cells. So again, this seems to demonstrate a bit of a planned response by the cells using phospho-tau against viral infection. Again, we got amyloid and tangles. We can't forget about neuroinflammation. There's a growing trend in the field that neuroinflammation may actually end up being the most destructive part of the entire process of the disease. And how we thought about this is, again, going back to amyloid, antimicrobial peptides are not just expressed and desired to go into as nets or attachment to the pathogens. They can also interact as a communication network, and they can influence the response of the cells and the tissue around it. So our approach here was pretty simple. We had AD or control cell cultures, and we exposed them to HSV1 and then looked to see how the response was different. So in these areas, it's AD versus control. So basically the genes that you're seeing here are altered by the presence of amyloid. So this is kind of like standard amyloid Alzheimer's disease response. The genes altered here are genes that are altered with the presence of HSV1. So it's two control cell lines, one with, one without HSV1. The genes here are basically how does amyloid respond in the presence of a viral infection. So both the AD and the control lines are virally infected. So that's our baseline. Now, when we add amyloid, what's the difference between? And interestingly, you can see there's a handful of unique downregulated genes, a very small handful of unique upregulated genes, but the majority of the genes are actually shared with the AD versus control. And what this tells us is that the presence of HSV1 has a very limited impact on how amyloid alters the AD gene pathology. But, and a huge but, this last section here, our baseline is both AD cell lines, one with, one without herpes virus. So here we're looking at how does the AD presence of amyloid affect the movement and gene expression of the virus. And you can see, as expected, most of the HSV1 specific altered genes are shared amongst these two groups, but there is a group of genes that are unique. These genes represent genes that are only occur when you get this presence of AD and virus together. And when we took a closer look at these genes in this unique section, that this idea of amyloid impacting the innate immune response, just flashing lights came up. NF-kappa B signaling, neuroinflammation, death receptor, TNFR2, STAT3, all of these are major inflammation and pathogen response pathways, and they're unique to when we have the presence of both HSV1 and amyloid together. So we've started to expand beyond this simple idea of a misfolded amyloid leads to tau, which leads to neuroinflammation. When we have the presence of a viral infection, amyloid, maybe it's forming plaques, or if it doesn't capture the virus before it gets to infecting the cell, we're starting to see, tau acting as part of this immune response, or amyloid potentially impacting or amplifying this response. We see amyloid attaching to the cells themselves, potentially altering the expression of the inflammation response, or leading to opsonization of these cells or apoptosis of the cells. It's created a much more integrated story. And I like to call this the beginning of the third age of AD research, when we stop kind of looking in this linear pathway and we start seeing today, I talked about tau as an AM. I talked about the immune modulators and the pathogen etiology of AD. Other parts of our group are working on a brain-gut axis and the roles of this amyloid peptide and its impact on your gut microbiota. Amylin is an ant. Amylin is expressed in the pancreas, is connected to diabetes, and that it may have an important role in infection. And A beta 42's ability to target endotoxins. So there's a much bigger picture. And I got like 2 slides left. And I want to try to connect this to a more global picture. And again, some of these diseases were even talked about today. We kind of have a little bit of like the early research, and all of these diseases affect some part of the central nervous system, and some of them are more clear-cut than others. And Dawson's disease, progressive multifocal leukoencephalopathy, that was, the disease was kind of sorted out in the mid 60s, and by the 70s, we had the virus targeted as exactly what it did. It was very easy for some of these it's a one pathogen disease, and once we found it, we could grow from that. But we're a bit into these later kind of more complicated diseases that it's not one pathogen. We haven't just found one thing that works for them. And chronic fatigue syndrome, we have a laundry list of possible pathogens that have been associated with it. Alzheimer's disease, I went through a couple of them in here. Within the last 5 years, periodontal bacteria has grown as another potential target. Parkinson's disease and its connection to the gut and the vagus nerve is growing. Multiple sclerosis was pretty much settled. That people first thought it was measles, but it was kind of settled that it wasn't pathogen associated. And then around 2010, we got a genetic study that was like, well, genetics can't describe all of it. So now that's blown up again. So where does that leave us? We're all involved in these research projects that are looking at highly complicated, complex diseases that are just not clear cut. In reference to my own research, we're looking at polymicrobial infections. Maybe it's not just HSV1. Maybe it's a combination of HSV1 and something else, or maybe it can be multiple pathogens. It doesn't have to be. It's the interaction with the immune system. We're getting complicated cross interactions. So HSV1 is affecting the immune system is affecting HSV1, but then we've got A beta 42 as a pathogen, or an A beta 42 peptide affecting HSV1 and also affecting the immune system. And complex genetic interactions, ApoE in Alzheimer's disease, a huge risk factor. But ApoE is also known to impact HSV1 infection. If you have the 4 carrier, you're more likely to have faster HSV1 infection. It alters APP production, and ApoE4 variant causes microglia to go into a more activated state. So I guess my point is that the clear-cut linear pathways of old kind of need to be thrown out the door, and we need to be thinking and exploring, as everybody here today so far has pointed out, in these not so simple pathways. So these are some of the mentors and contributors to these projects and the various degrees of funding that we've needed to make this happen.
Thanks, Will. You are completely speaking our language. That's exactly what we think.
Well, yeah, such incredible applicability to ME/CFS as well. In fact, that's why I sought out your lab, and also Rob Moore, who you knew I was also friends with. There's a autopsy study of the one ME/CFS brain that's full of amyloid. Polymicrobial infection is almost certainly so important for ME/CFS. I mean, just huge parallels. So thank you so much. It was an incredible presentation.